Okay, sorry, it's a bit of a hitch. Yeah, thank you very much, Len. So hello, everyone. Thank you very much for tuning in. My name's Lisa, and I just finished my PhD at the University of Edinburgh. And as Len said, I'm just going to present a piece of work that I've done for my thesis. Although this is really the story of how a relatively straightforward meta-analysis went way out of hand and transformed into this monster of a model, which ended up being a really interesting piece of work. So I'm quite excited to present the results. So I really hope you enjoy it. So a little bit of background here. As you might know, foxes are now living among us. You might be familiar with this phenomenon if you live in Europe. They're pretty much everywhere. However, it's becoming now more of a global phenomenon. And so more and more reports of urban foxes are around the globe. This can be a problem since foxes can actually carry quite a lot of parasites, some of which are of quite high zoonotic importance. Think, for example, rabies, or more recently, the Echinococcus multirocularis. And so it is really not that much of a surprise that there is a pretty substantial amount of work done into try to determine if living in urban environment has an effect on the prevalence of these parasites. And I'm going to give you a very quick tour of what the urban disease ecology of foxes field looks like. And to do this, I'm going to present three talks, which is obviously not a comprehensive look, but I think is representative of what this field actually looked like. The first of these papers was done in Bristol as a study conducted to see if the prevalence of mange changes between urban and rural foxes. And what they found was actually foxes in urban areas are actually at a higher prevalence of mange than rural ones. The second one was in Switzerland, Geneva to be precise, and they looked at the entire gastrointestinal parasite community and found that the Toxoscaris leonina was actually had a lower prevalence in urban environment. However, they did not find any other statistically significant difference between the two for the rest of the parasites that they looked at. Finally, their study was conducted in France. And they look at the prevalence of the Echinococcus multirocularis, which is one of the highest zoonotic threats in Europe at the moment. It's quite serious and causes a pretty serious disease called alveolar echinococcosis. So it's very, very much studied. And what they found was that the prevalence of the Echinococcus was lower in urban areas, but also higher. They actually looked at two different towns and found contrasting results. So I hope I gave you like a good idea of why it's so difficult to actually find out what the overall underlying effect of urbanity is. Partially, this is due to the fact that, as I mentioned, foxes carry a lot of parasite, and different parasite species can respond differently to urban environment. But it's also due to the fact that it's really difficult to determine and define what urban is. And so drawing meaningful comparison between different cities, different towns, becomes really a problem. And so this brings us to the question that started this whole thing, which is like, I wanted to determine what is the overall effect of urban environment on the fox parasite prevalence. And to do this, I had a really simple two-step plan, which will be collect a lot of data and then analyze this data using the same objective measurement of urbanity. So to collect my data, I started as I said, as a quite normal meta-analysis, went to Web of Science, selected the papers, then selected them further to according to a number of criteria. And my final list included 312 papers. So I went through the papers starting to extract the data I needed, and found out that few, if any of this, actually reported coordinates or location at the fine scale that I actually needed. which will make basically my analysis impossible. So what I actually did was to extend the collection period by writing to the authors of these papers and personally requesting the raw data of their data set, which would include either their coordinates or the location of the collection of each data point at a lowest possible local scale. And this yielded my final data set, which included almost 73,000 data points. And this is what the data set looks mapped on the map of Europe. And as you can see, this data set spans 20 years and 27 countries, and is obviously not regularly distributed, neither in space or time. So a model to incorporate this will have to have a spatial and temporal explicit terms to account for this. I also was also very fortunate, and over 30 research groups actually agreed to collaborate, which came though with its own set of challenges, because obviously this means that different groups would use different methods, detection methods, and we work with different samples. So this will also have to be controlled for in the final model. You probably already noticed, but there's also almost 70 different parasite genera in this data set, which would make basically incorporate them straight into the model almost impossible. It would be impossible to have the power to draw any meaningful results, let alone at a species level. So what I ended up doing was actually classify this parasite according to a set of functional traits. In particular, use transmission pattern and parasite type. For the transmission pattern, you will have the closely transmitted parasites, which are airborne and contact-borne. The non-close, the ones that persist in the environment until a new host come and pick them up. The intermediate host, which are the ones that require the intermediate, which require an intermediate host, and the vector, which require a vector to transmit. Parasite type is defined as macroparasite if the parasite reproduce within the body of the host, so all the helminths and ectoparasites, and microparasites, which includes all the parasites that replicate within the body of the host. So now that we have collected our data, we have to find this objective measurement of urbanity, which I mentioned is a bit of a problem. Fortunately, in recent years, a number of remote sensing data sets have been developed, and I am going to present two of them, both developed by NASA. The first one is the H-base, which is a measure of the percentage of built up space within each 1 square kilometer pixel, which goes between 0 and 100. The second one is the human footprint, which is a composite measure of the pressure that human put on the environment, and goes between 0 and 50. If you plot these two together, you have your own map of urbanization level, and then you can just plot your points, and you will have like the score for each of these two data sets. Actually, initially I was thinking of combining these two to have one single measurement, but I never really found a meaningful way to do it. So I ended up fitting two models with each of them. I'm only going to present the H-base one, but if you want to know about the human footprint, please just let me know. Also, if you have any suggestion how to effectively combining this two, please just, I'll be glad to hear it. So now that we got our data set and we have our measurement of urbanity, this is the model that I fitted. This is a hierarchical spatial and temporally explicit model fitted with INLA, which is a Bayesian method, which provides quite a lot of advantages compared to MCMC, since it's much less computationally heavy. The response variable was Bernoulli binomial distribution. Is basically the answer to the question, is this fox infected with this parasite? And you can see it has like, it had a bunch of linear effects here in green, which control for environmental, geographical, and parasite covariates, which could influence the overall probability of infection of foxes. In red here, you have your random effects that control for heterogeneity in the data set. and your spatial and temporal effect. And in blue, you have your nonlinear effects, which are the interaction covariates, which are the real answer to the question that I posed, which is the interaction between the sorry, the interaction between the urbanization level and the parasite traits. In particular here, this model I'm going to present has the microparasites. Since micro and macro are mutually exclusive, I could only fit one at a time. And then you have all the 4 parasite transmission traits here. So I should say that these are fitted using a random walk of the 2nd degree to account for non-linear effects. So these are the results. On the x-axis, you have your urbanity level between 0 and 100. On the y-axis, you have your probability of infection. And these are the 4 transmission patterns, transmission traits. And you can see that there is really not any distinguishable pattern there. However, if you actually look at the microparasite, you can see that there's a very clear pattern of increased probability of infection that peaks around 70% of built-up space, and then reduces as built-up space percentage goes up again. And these actually follow quite closely what you can see in the population of fox density actually is, because this is roughly suburbs, which is the optimal environment for foxes to live in urban environments. So to conclude, the transmission pattern did not seem to be important in determining the parasite response to urbanization level. However, there is a measurable nonlinear effect of urbanity level on the microparasite, which follow closely fox densities in city. And with that, I would like to acknowledge all the people that actually have sent me data. There's obviously not been possible without them. My supervisor, funding body, research group, and obviously you for your attention. Thanks.